Om Namah Shivaya. Hi friends, how are you? In this video we are going to learn about metabolic factors that control the food intake. This is one of the function of hypothalamus. Metabolic factors. First, plasma glucose. Plasma glucose concentration is important metabolic factor for regulation of feeding. Discharge of ventromedian hypothalamus. Discharge of ventromedian hypothalamus, we call this as satiety center, partly depends on its glucose utilization. Following feeding, plasma glucose concentration rises, that in turn increases the activity of neurons of satiety center. Following feeding, plasma glucose concentration rises, that in turn increases the activity of neurons of satiety center. Increased discharge of satiety center inhibits the feeding center. Increased discharge of satiety center inhibits feeding center, that finally inhibits food intake. Thus the person feels sated. Food intake remains inhibited till load glucose concentration remains high. Food intake remains inhibited till load glucose concentration remains high. This is called glucostatic hypothesis of regulation of food intake. Hypothalamus, by sending plasma glucose concentration, not only regulates food intake. Hypothalamus, by sensing plasma glucose concentration, not only regulates food intake but also indirectly regulates plasma glucose concentration. Therefore, hypothalamus is called glucostat. Remember this, it is a glucostat. Hypoglycemia is a potent stimulus for food intake. Next metabolic factor is malonyl-CoA. Malonyl-CoA accumulation in the tissue inhibits food intake. Malonyl-CoA accumulation in the food inhibits food intake. Malonyl-CoA is produced from acetyl-CoA. Malonyl-CoA is converted to fatty acid by fatty acid synthesis. Malonyl-CoA is converted to fatty acid by fatty acid synthase. Therefore, currently the focus of research is to develop an agent which will inhibit fatty acid synthase so that malonyl-CoA can accumulate and inhibiting feeding. Malonyl-CoA can accumulate and inhibit feeding. Malonyl-CoA is also seen to inhibit hypothalamic neuropeptide Y synthesis. Malonyl-CoA is also seen to inhibit hypothalamic neuropeptide Y synthesis by inhibiting the formation of neuropeptide Y mRNA. It also induces weight loss and decreases fat storage. Malonyl-CoA also induces weight loss and decreases fat storage. Next, amino acids and fatty acids. Among the chemical signals that give rise to satiety are certain amino acids and fatty acids observed from gastrointestinal tract. Among the chemical signals that give rise to satiety are certain amino acids and fatty acids observed from gastrointestinal tract. Next, body temperature. Decrease in body temperature increases food intake. Increase in body temperature inhibits food intake. This is called thermostatic hypothesis of food intake. This is called thermostatic hypothesis of food intake. Therefore, anorexia, that means inhibiting of food intake. Anorexia is a common feature of fever due to any cause. Okay, anorexia is a common feature of fever. Anorexia is nothing but it inhibits the food intake. Okay, in fact, change in food intake changes body temperature by changing body metabolism and vice versa. Change in food intake changes body temperature and vice versa. Okay guys, clear? If you like this video, like, share and subscribe. Thank you.